So what I thought might be interesting is just to give an example where tools that came from physical science and some thinking that comes from physical sciences, that comes from mathematics, can be used to address a problem, in biology and physiology, and also in a way that was really unanticipated for us at the beginning, ends up having some impact on health, right? I'm not in the medical school. I've had my share of broken bones and arthropods, but I try to stay away from hospitals. But sometimes it's just interesting, right? To find out that you're doing something totally useless, and then lo and behold, it turns out to be very useful. So I think that's so this is sort of, I'm just going to go through sort of a story, and I'll make sure I cut it off by 11 o'clock or so not everybody goes crazy. So what are we looking at here? Anybody have an idea what we're looking at? Nice pictures. It's good to have pictures. I tell you, the competition for covers of journals now is fierce. It used to be easy. So this is actually a chunk of, these are blood vessels. They're candy-colored blue for venules, veins, and red for arterioles. This is a piece of the cortex, mouse cortex, which we filled every microvessel, every single vessel with a dye, with a gel and a dye. And we reconstructed the vasculature. And we did this because we wanted to understand basically the physics of flow. So this is a piece of reality from the mouse. And at the end of the day, we want to make a little model with resistors and capacitors and symmetry, something we can understand, and then something we can use to predict some other independent quantity, which is actually going to be the patterns of flow, as we'll do sort of a perturbation experiment. We'll go in there and squeeze on a vessel and turn off the flow and see how this defect propagates. Now, these are very tiny vessels. There's no scale bar here. Some of these vessels are down around 2 microns. The human hair, as they say, is 100 microns, which is enormous under the microscope. And these little mountains, it looks like sort of maroon bells gone wild, are maps of activity, sort of electrical activity in the brain superimposed on top of the vasculature. So there's going to be a lot of techniques involved in having gotten this far. And so I'll try to introduce different techniques we use, all in the guise of trying to understand how blood flows within the brain. Okay, so then why might you guys be interested? Why did you trudge all the way to La Jolla? And I mean, it's great. The ocean is beautiful. Actually, my daughter's giving kayak tours if any of you want to go down to La Jolla shores. But there's a couple of interesting issues that come up in just thinking about blood flow. One is it's a resource allocation issue. There's a finite amount of blood within the brain. There's actually not enough blood that flows in the brain to address all the energy needs if every neuron in the brain started firing at its sort of maximum rate, or close to its maximum rate. So you have to shuttle this resource around, right? So it's interesting. It's like an economic issue, you might say. There's issues early on in development. Neurons and blood vessels grow together, many of the same signaling molecules, lineage molecules that cause cells to sort of migrate in a certain direction, also cause the vasculature to grow. There's clearly a correspondence between energetics and neural activity. So speaking of Bell Labs, Seiji Ogawa, who actually had his office next to mine at Bell, he's a good friend. He was in Kamakura, Japan now, must have been the 10,000th person to try to do magnetic resonance imaging on rodents. And one day, the signal just started to drift, okay? And it just got brighter. And, it would have been just too easy to say, I don't know, something's wrong. But he traced it. He wanted to understand why. Now, Seiji was trained in sort of physical chemistry. And what he knew that the magnetic properties of hemoglobin, just like its optical properties, change with oxygenation. So when oxygen is bound, it's diamagnetic. It lines up the spin on the molecule, tries to line up opposite to applied magnetic field. And when there's no oxygen in the deoxygenated state, it's paramagnetic. It tries to line up with the magnetic field. So the resonance condition is different. So we discovered that the resonance condition of hemoglobin, that the magnetic properties of hemoglobin, will change the resonance conditions of nearby water molecules, or equivalently the relaxation rate, the spin relaxation rate of nearby water molecules. And this has turned into an industry. Well, people will basically look at relaxation rate of water in the blood, due to changes in the magnetic properties of hemoglobin. And if there's a lot of neural activity, hemoglobin is going, at first when there's a burst of activity, it goes from sort of net oxy to net deoxy, and then there's some complicated overshoots, which I'll ignore for the moment. But there's this interplay between energetics and activity, and functional MRI, fMRI, Seiji's invention, is sort of the key tool to probe brain activity within people. So you want to understand, actually, the signal is easy to interpret in some places. It's very hard to interpret in other places. It's a little controversial still. So you'd like to understand blood flow because you'd like to understand how this match is made between energetics and activity. And then, of course, there's the standard line you give anytime you go to the NIH and beg for money, which is that there's a connection between vascular disease and neurological decline. Okay, so the other thing that I think was explained, you don't really do anything yourself today in science. You have a group of people work with you. This is my lab. This is from last summer. It's a little dated. That's the fountain in Revel if you walk over to the other side of campus. And I'll start to identify who's done what. Okay, so let me identify a couple of features on the vasculature. There are these big, well, not that big, maybe in the mouse, about 30 microns. There are vessels on the surface of the brain, which we'll get back to. And the surface vessels on the brain, which are sort of the key suppliers of blood, have these stringers that drop down. These are called penetrating vessels. They take blood from the top, and they go into sort of the bulk of the brain, and they feed these microvessels, this sort of mesh of vessels. So, since most of you are sort of mathematically oriented, you want to think of it that the surface vasculature, as I'll show you, is going to form a network, like a road system in 2 dimensions, kind of like Manhattan. The subsurface vasculature just forms this sort of mesh, this 3-dimensional cavern, which you can, as I'll show you, you can go from any one point to any other point. And then you sort of have these 1-dimensional features, supplying blood and moving blood away. And I'll get back a little later as to how we actually did this. So the first question is, what is the nature of blood flow on the surface? So in order to do this, we took, this is done in rodents, both rats and mice. And the interesting thing from the point of view of studying blood flow is a mouse is like a truncated rat. It's not even a small version, it's just a truncated version. And what we did is we filled all the vessels with a dye that had a fluorescent color to it. And we mapped out all the vessels. This is just on the surface. And it looks like these vessels are ending here. This is because they're going to plunge downward. But then after we sort of filled out mapped the whole surface, what we did is what I connected here in black are all the places where the surface forms loops. So you can think of, if you're feeding something, normally if you're plumbing your house, you're plumbing a building, you think of sort of a root system or a tree system where branches branch and branch and branch, which basically means there's only one path to get from where the original flow comes in until you get to some point. But here what's happening is, suppose you want to deliver blood to this point. There's a path that's this way. There's a path that's this way. There's a multitude of paths to get blood from one point to the other. So there's a kind of redundancy that nature picked up that allows flow, in fact, to take many different paths. And also what you could envision is if you were at a point here, and all of a sudden the brain tissue under this region needed a lot of blood, you can imagine flow coming in from every possible direction, which again can never happen in a tree structure because there's only one path inward. So even just a simple reconstruction like this, a simple map, gives you a lot of ideas. So the lesson from this is that these surface arterioles are arranged in this very interconnected network. And let's see if it robustly circulates blood. So that was a static picture. So now we need a couple of pieces of technology to measure blood flow. Well, Winfried Denk, who's a close colleague, when he was a graduate student at Cornell in Watt Webb's lab, invented a technique called two-photon microscopy, which is very, very popular in cell biology and in neuroscience. And in a nutshell, you often want to scan deep into tissue like brain tissue or even your finger, which is very scattering. And you need some technique, actually, in which you can excite molecules at a point, and you're relatively insensitive to the scatter. So what you might remember from an optics class is light comes in a sort of a focus bundle, like a conic, and then it forms sort of a waist. Okay, that's where the focus is, and then it goes down. But basically, you're exciting the same number of molecules at every possible plane with linear absorption. If you have a nonlinear process, which means very, very bright light, so that you can get molecules to absorb 2 quanta instead of 1 quanta, then in fact, the only place where the power is, in this case, power squared, 2-photon absorption, is the strongest, is right at the center, is right at the focus. So we put light in, we're scattering tons of it away. But in fact, the scattering lengths are about 100 microns. You lose about a E-fold of light every 100 microns in the brain. But that's still very long compared to sort of the wavelength of light, which is around a micron. And this enables us to image deep into scattering tissue. So what we're going to do is there's blood flowing in these vessels. And we're going to do a particle tracking. So I'm going to scan a beam along a direction. And every time it goes through a blood cell, the blood cell is dark, fluorescently dark. There's no intensity. Every time I go into the serum, I'm going to put some dye in the serum. It's bright. So I could just look at bright and dark fringes go past my line of light. And I could also measure diameters of blood vessels. And I could do things like the data shown here. As a function of time, I can map the diameter of a vessel. I can map the speed of the blood. And the critical thing, I could ask how many blood cells per unit volume go by in a certain amount of time. The flux is like electrical current. And I had this nice calculation. So right here is actually stimulated the forelimb of the rodent. And I get this, not surprisingly, big spike in flow. So this is a tool. I can measure dynamically. I can measure speed. And so let's just think about what happens in the diameter. So this is an experiment done with Anna DeVore when she was a visitor in the lab. She's now a assistant professor here at the medical school, associate professor, sorry, at the medical school. And I'm looking down at the cortex of an animal. And one of the special things that just happens, it didn't have to be this way, but there are areas where, these are called primary cortical areas. This is where sensory information from the outside world comes in. It could be auditory, it could be visual, it could be tactile. And there's sort of a map of the sort of sensory sheet. So if it's the visual world, it's sort of what's called retinotopic map, different positions in space, go to different places on the cortex. If it's tactile, then different places on the skin go to different places on cortex. So we're in sort of this tactile or somatosensory cortex. And if I put an electrode here where the blue circle is, and I'm touching the forepaw of the animal, I get a lot of neuronal activity, multi-unit response. It's a very gross measure of neuronal activity. And if I move a little bit off, that's this blue spot, that's this red region called the surround. I get essentially no activity. So I get a very focused, activated spot. And this is done on an anesthetized animal. Now, anesthesia is a very unnatural state. It's not sleep. Emery Brown, actually a colleague of Todd's, colleague of mine, really worked out very nicely what's happening during anesthesia. It's more like a seizure state than a sleep state. But we're using it here because it keeps the neural activity very, very focused. Now, at the same time, I could use this previous technique, this previous two-photon technique, to look at the diameter of every blood vessel. And let me just walk over here to sort of the composite data. So this is what I see. So right at the center, the blue center, where the neural activity peaked, I have a stimulus, and the vessels dilate. They get bigger. So there's more blood flow. But if I walk away, and this is one example, but this is now average over many examples, and I go here to the surround, there's a little bit of an increase, but then gradually there's a rather big decrease in the diameter of the vessels. So you're basically pushing blood towards the middle. So this idea that you have a fully interconnected set of pipes that can push blood where it's needed seems to pan out. This is what biology seems to do. So think of it again. We use technique from physics, a microscopy technique from physics. We use perhaps some very naive thinking from physics to motivate the experiment. And now we've learned something one thinks about biology. So blood flow on the surface network is dynamically distributed. And then we want to know what happens if there's an occlusion. So suppose you're driving, even in an insane place like Boston, okay? And someone blocks a street. I mean, are you stuck? Well, if the streets are connected, so you think you can go around at least one blockage. You know, I grew up in New York, and I mean, I think maybe 1/3 of the streets were blocked at any given point in time. I mean, there was always somebody putting a traffic cone in the middle of the street. God knows why. So we want to do, this is sort of, again, a very standard idea that comes from the world of physics and chemistry, is to do a perturbation experiment, perturb the system, see how it reacts. You know, as the perturbation gets weaker and weaker, we expect linear analysis to hold. So we need a way then to block flow. So again, we're going to appeal to optics. In this case, we're going to appeal to linear optics. So these are vessels on the surface. It's kind of street direction of moving blood. And if I want to block flow, what I'd like to do is squeeze down on this vessel. So there's a couple ways to do this, and I'm just showing one way. This is actually a little bit dated now. This is the work of Chris Schaefer. Chris is now an associate professor at Cornell at bioengineering. And actually, Nozomi Nishimura is also an assistant professor at Cornell in bioengineering. Actually, people have done well. And Lee Schroeder is an assistant professor at Michigan. So our idea is that we put in a molecule that when you excite it with light, makes what's called a free radical. It makes a reactive species, right? Like a free electron species that'll just glom onto what's ever there. And in time, this will cause proteins to coagulate. And this could cause a blockage to form. And since these vessels are at the surface, what we do is we come in with light. This molecule has a funny name. It's called Rose Bengal. And we come in with light just at the threshold, right, for causing damage. And then as the light goes through the vessel and scatters in the tissue below, it's sort of below threshold for causing damage anymore. And the consequence of this is what's shown here is that this dark spot is where there's no longer blood flowing. The bright spot is because the plasma in the blood, the liquid part of the blood, is filled with dye. And the red blood cell count is relatively low in this region, so I basically backed it up. And of course, the blockage at this point. So what this means is using optics to our advantage, we can measure all kind of parameters, all sort of physiology in the system, come in, make a perturbation, and remeasure it, and know exactly where the perturbation is, right? And so the experiment that Chris did is as following. We mapped the entire vascular surface just with this sort of wide field two-photon technique. And then from this seemingly complicated map, which contains veins as well as arteries, we just extract out just the veins. And you can see very nicely just the arteries. You can see this loop structure very nicely. And then we're going to ask what happens when I make a block at this point. And in fact, does the downstream flow come to a halt? Or does the system just rearrange its pattern of flow? So that's shown here in this one example. So this is again, this is sort of a traffic circle of blood flow. If you drive in Boston, it seems totally natural. And the numbers there are the speed in millimeters per second of the flow. And we've mapped as much as we like. And then we come in and we make a blockage at this point marked with an X. And then we can take a full image afterwards just to confirm that the blockage was very localized. And what you can see is that speeds have changed, but the downstream vessel always reverses its direction. Okay, so initially it was flowing at 3 millimeters per second in one direction. Now it's flowing in 5 millimeters per second in the other direction. There's this rearrangement of flow, but no vessel goes without flow. Okay, so the system just rearranges to the blockage in one pipe, if you will, one surface vessel. And in some sense, it's robust. And the magnitude of the flow is essentially unchanged. So this surface vessel not only allows blood to flow in from many directions, which is probably its chief reason for being, but it also, consistent with sort of simple physical ideas, okay, allows the redistribution of flow. And you notice that this lattice I showed you before, these aren't regular. If these were just perfect hexagonal lattices, you can imagine just getting points where pressures exactly cancel out. But it's kind of the irregularity that always guarantees there's going to be some flow after a blockage. Okay, so what we've learned is that the surface network is robust to these single occlusions. We could go on, but let's now move to a place where we're going to see a much different answer, and it's also going to sort of stress our technical capabilities a little bit.
Dave, brief question though. That result is fascinating. Is that a consequence of evolution? Is there an adaptive mechanism going on? Is it, there's this inherent robustness and sort of quote unquote intelligence based upon the sort of the surface structure, the topology? What is the best interpretation?
I just think it's because of redistribution of flow. I think that the fact that it's insensitive to a defect, I think is secondary to the fact that you want to redistribute flow. I think the best way to think about it is the surface is it's like a hexagonal lattice, like I mentioned, with some disorder.
Right.
And I don't think we're living too long these days. I don't think nature built this thing to be insensitive to defects. I think it did build it to respond very rapidly to heightened neural activity. You just don't want to build up metabolites and stuff in different regions. That's a guess. Now, it's a great question because you could ask, how do you test this? You need an alternate sort of vascular design, and it's hard to plan this, but sometimes little things happen by accident. So Randy Johnson, there's a professor of biology, was here. He moved to London some time ago, and he made an animal that just happened to express this protein ApoE. It causes vascular growth. He did it in a kind of brain cell called glial cells. What wasn't the least bit anticipated is that it had an entirely different vascular plan. There were no big vessels, small vessels. It was just one mesh. The animal lived. And we did some preliminary measurements, but it'd be nice to go back and compare it with these alternate plans. It'd be pretty wild. Yeah. So I want to turn the attention now to routing of blood from the surface network deep into the brain. And now we need a couple of tricks. We need to do two things. We're going to get deep into the brain. We're going to need a trick to sort of do this reconstruction. I showed you this pretty picture at the beginning of this whole reconstructed anatomy. Well, there's a couple ways to do this. I could take a brain and I could slice it really thin, like prosciutto, the delicatessen, and then you can have somebody mark it and analyze it and align the sections. But sort of the last decade or so has been this huge push within neuroscience, continuing push, I would say, with open problems for automating anatomy, right? It's ugly. Things are interconnected. It's sort of like the way when you're a little kid, you build electronic circuits. All the wires are all over the place. It's impossible to understand it. And then you just sort of want to stretch it out and make a simple wiring diagram. But the first thing to understand how nature wired things up or plumbed things up is you have to get a full reconstruction. So we had an idea, and we wanted to, instead of using a knife to cut, we wanted to use light to cut. So we could image and then cut and image and cut. So coming out of Lawrence Berkeley Labs in the mid-'60s was a lot of work using a lot of amplified ultrafast light. So these are pulses that are maybe 100 femtoseconds in time. So they're broad. They're almost a little bit white, right, because they're so short in time. And what happens is when you focus these, you can amplify light to the point that if you focus it in the middle of glass, which you think of being transparent, the electric fields are on the order of the electric fields that'll strip electrons off atoms, right? The binding energy is about 10 volts per angstrom. That's the sort of the natural electric fields. You could easily get up to a couple of volts per angstrom, just in the privacy of your own laboratory. And this is the same Chris Schaefer. This is part of his thesis work, just focusing into glass in Eric Mesur's lab. And what's very bright there is just the light as the electrons fall back into place. It's just the recombination light. So we're going to use this as a technique do histology. So we're taking something which is sort of way out, originally was way out on the fringes of optical physics, and we're applying it to anatomy, which seems ever as messy and watery as you could ever get in biology. But the idea is simple. We could use our two-photon microscopy to image vessels, which are labeled with a dye, as deep as possible. And then, by about 150, 200 microns down, we're running out of signal to noise. It's just too much scattering. And then we come in with light, and we just vaporize off about 150 microns of material with good accuracy, micron accuracy, and then we image again. So it's image, dust. Image again, make more dust. Image again, make more dust. And at the end of the day, you end up with this reconstructed vasculature. Okay, so you have fluorescent intensity. You do some, now classic mathematical techniques. Match filtering came out of World War II with tubes. We end up with this vasculature. There's imperfections. There's places the fill was not quite there, so looks like there's gaps. This is sort of purposely typical Wirtz data. So then you have to do a little bit of computer science to say, well, it's really unlikely that there was a gap. So we try to automatically recognize the gaps and then go back to the data and see if, in fact, there was some faint fluorescence in this region compared to the outside. So a lot of data analysis, and still today, is you have a computer do as much analysis as possible and then sort of highlight the difficult spots, and you go back there by hand and cross-check. So at the end of the day, you end up with one of these pictures of the vasculature. And let's use this to our advantage. So that's a pretty picture. And each of those branches are, like I said, we use a matched filter. It's just like a little segment. It's not a really important detail. But we want to abstract this to something we could put our hands on, which is electric circuits. We all know Ohm's law. We all know Kirchhoff's law, that current flows in and current flows out. And it's conserved, unless you're a bank and you take 1% or 2%. But in physics, it's conserved. So what we want to do is actually take this ugly reconstruction and turn it into something that we could deal with mathematically. So there's a vertex. And nature was very kind. It always made these triplets, only triplets, right? And this is a piece of pipe. And it has a diameter, and it has a length. And that means it has a resistance. So we're turning biology into an electrical network. So let's just get a little bit of some of the features that we could pull out of this. We just want to do a little bit of analysis. So on the surface, we looked at the surface, and we looked at how many edges are in a loop. If it was hexagonal, we'd have 6 edges and a loop on the surface. It's not quite perfect. It's about 5 edges. That's this thing labeled PO vessels. If I look deep, what I always see is the following. I could just start wherever I want, and I could ask, how long does it take to come back to where I started from? Okay, and I make these loops. They peak at around some 7, 8, 9 edges or so. These details aren't important. They're always small, right? It's not what you don't just have a wire that goes off a very, very long distance, what people call small world networks. Okay, you're always very local. So by doing this kind of quantitative anatomy, rather than just looking at a few sections and saying, oh, there's small vessels, big vessels, but by reconstructing everything, you could understand something about the topology. And it's all local roads. Okay, once you get deep, there's no superhighways, there's no tunnels, there's no hopping from hub to hub. It's all very local. Okay, so one of the questions is, if it's all very local, right? Let me play this same game, and let me try to break one of these vessels and seeing if these local roads actually support reperfusion. How important is one vessel? That's the basic question. So we want to do this, and I want to block a single vessel, but there's a problem. Now I'm deep in the brain. And before I used this trick, I used linear light, right? I had it just at threshold. There was nothing on top of me. It's just air, right? And then there was brain tissue. Now, in this cartoon, I'm plumb in the middle of the brain. So I have to have some nonlinear optical technique in which the intensity is too low to cause any damage away from the focus, And at the focus, it depends on the absorption of many quanta. So it's a very, very nonlinear technique. And then I could cause some damage at this spot. And then away from that spot again, there's no more damage. So this was the thesis work of Nozomi Nishimura, as I mentioned, is now at Cornell. And well, if you come in with a little too much energy, so what we're doing is we're focusing right in the middle of a vessel, just like Chris Schaefer had done in the middle of a piece of glass. Same technology. And I create, actually, by putting that much energy right in the middle of a blood vessel that's like 3, 4 microns in diameter, I create a little cavitation bubble that just expands out. And this cloud is just the cloud of dye that was in the vessel. You might say this was a little too much power. You had too much power. At some point, you have no more rodent. You have so much power, but we won't go there. But if I'm very careful with how I adjust the power, I could put in literally a small number of optical pulses. And what I've done is I've just damaged the vessel, and I've stopped flow within this vessel. And the brightness here is just sort of serum building up, labeled blood plasma building up. So I could really block one vessel. And the consequence is kind of interesting. So what we've done is we've blocked one vessel where the X is, and the labels like D1 are one vessel downstream, 2 is 2 vessels downstream. So what happens is things really don't recover directly downstream, like on the surface. But by the time I go like 3 vessels downstream, I'm back to where I was. Okay? And the critical thing is if I do some kind of classic marker for neuronal death, there's no neuronal death. So killing one of these little microvessels does not hurt anything. And this larger network, maybe not one vessel downstream, but a few vessels downstream allows flow to recover. So there's a redundancy now, not just on the surface, but also within the depths of the brain tissue itself. Okay, so this is work from Andy Shih, who's now a professor at Medical University of South Carolina, where we did exactly this technique. The little green dot is where we damaged a vessel, and the point of this picture is the white dots are marking healthy neurons, and there's really no change in density at the point of damage. So these are sort of classic anatomical tools, histological tools, look for cell death, that are sort of critical that go together with these sort of high-tech tools, right, to perturb the system. Okay, so the other issue is, what about the long range? I mentioned before that we didn't see anything in our pictures, in our analysis, for long-range connectivity. But let me be a little more stringent about this. This is actually a really important point. If there's some long-range connections, if vessels in one place can communicate with vessels in another place. And I'd like again, it introduces a little bit of physical thinking here. So I'm really asking if I could be in a microvessel here, and it just happens that it has a couple of shoots over to this region. Okay, so let's see how this goes. So the idea is, let me just remind myself of something here, is the following. We measured all these distances and diameters for the microvessels between vertices, so I could convert this length and this radius into a resistance. Okay, and this is basically a lookup table, and what this means is it's this black curve. The dashed line is the curve I expected if I had this sort of beautiful parabolic flow, like laminar flow. Because there's red blood cells, because I'm not just pushing liquid, but I'm pushing red blood cells, the flow is a little different. It's actually an opportunity. It's a real bioengineering question. So what's happening is that the vessels are just about the same size as the red blood cell. So at first, when the vessel is just a little smaller than the red blood cell, the fluid right around the blood cell acts as a lubricating layer. Okay? And this was a problem solved by Sir Lighthill, that you actually, if you want to push rocks through a pipe, which people do with coal slurry, right? Then the pipe is not that much bigger than the size of the pieces of coal, but the fluid acts as a lubricating layer. But then the problem is when the red blood cell gets just a little bit bigger than the pipe, then all of a sudden it has to buckle, and the resistance just starts to explode. So that's the origin of this resistance curve. Okay, so we use this as kind of a lookup table. And then what we could do is we could say, well, let's ask what the resistance is between a point here and just all other points as I start to walk away. So I'm just basically, on the computer, going to do sort of an Ohm's law experiment, where I'm going to put a potential, and then ask what current flows as I probe from here to here, here to here, here to here, here to here. And this is the idea. If I really did have very, very long pipes, okay, or if I had a structure where I had this sort of interconnected vessels, and then they just formed a little community on their own, and then there was another little community next door with just one or two connections between them, and then another community next door with just a number of connections between them, sort of like gated communities in Southern California, right? A lot of roads, but only one or two roads between communities, right? Then the resistance would just grow with length, right? As I just stack resistors in a sequence, the further I get apart, the higher the resistance goes. If there's a two-dimensional network, it goes like the log of the resistance. It's still a function of the distance L, but very weakly. But if things are truly a mesh, a three-dimensional mesh, what's happening is as I'm going further apart, it's true the direct path has a resistance that goes up, and now I'm adding all these other paths, right, in parallel with my direct paths. So the number of paths I add in parallel, okay, compensate for the increased length of the direct path. So the resistance should go to a constant. And indeed, when we take all of our networks that we reconstructed, our 4 networks, and average over all possible paths, what you see is there's a lot of scatter. What you see is that the resistance goes to a constant. And you could just sort of follow this jaggedy black line. So this really shows that on all, we looked before in short length scales, now we're looking on long length scales, that this microvasculature is purely sort of an interconnected three-dimensional network with only short connections. And so now let's sort of get to this crux issue. So we have this microvascular network, and it forms this largely structuralist network in 3 dimensions. Okay? And what we want to know is the following. Is this network sufficient to support the lateral flow of blood throughout the brain? I mean, why do I have all these vessels on the surface if I could just push blood at one side and just have it flow through the whole network? And I showed you before, and I think this is the next slide. I'm just going to go back and forth, that there are these penetrating vessels that drop down from the surface. I'm showing here in red, and these are the return vessels in blue. So why can't I have blood just push through the whole network? Well, why do I need to have these feeder pipes every so often? And what we can do experimentally is we can go and we could block one of these feeder pipes. And we could ask if actually blood gets restored from the side. Now, the question is not so easy to answer because, or the reason you want to do the experiment, it's not easy to answer theoretically, is that I could look here. This is the flux of blood. These are measured numbers that come from these penetrating vessels. It's about a nanoliter per second. I mean, that's not the important issue. If I look at how much flux comes from these microvessels, that's this cloud. I mean, there's quite a spread in values, but the median value is about a hundredth of this, about a hundredth of a nanoliter per second, but there's a couple of hundred vessels from the side, so the numbers are kind of balanced. And when things are kind of balanced, you're kind of forced to do an experiment to find out who wins. So we're going to do this very similar experiment what we did before. We're able to measure flow within all these microvessels, and then we're going to come onto the surface. So this is a 2-photon image of the surface. These are the arterioles, and these dots are where vessels plunge. And now we're going to use linear light, because nature was very kind. It just had this little snippet that I could grab a hold to for these plunging vessels. I'm going to make a blockage here, and then I could confirm it. This is an image sort of looking in the Z direction, going at depth. I can see that I blocked this penetrating vessel very nicely. So this started again with a thesis work of Nozomi Nishimura, another project. And what Nozomi did was she found the penetrating vessel of interest and said, this is the guy I'm going to block. And And then she just found as many microvessels as she could within, sort of within about 200 to 400 microns below the surface. And she mapped out what their flow was, and she came back. She blocked this vessel, and she mapped out what the flow was again. And she looked at how the flow changed as a function of how far you were from this target vessel. And what she found is that blocking this penetrating artery just caused the flow to go exactly to 0. And it doesn't recover until you're a radius of about 250, 300 microns out. 500 micron diameter cylinder of death from blocking a single form of vessel. So blood is just not coming in from the side. Kind of a conundrum, okay? So this is kind of a serious issue. And what it says is that this class of arteries, called these penetrating arteries, these are a bottleneck in the supply of blood to the subsurface microvasculature, okay? So I'm going to skip a little bit, but just want to make a point here. We could actually image this damage. So that was shown. Those are what's called an acute experiment. We did the experiment. We looked at flow. We're looking a couple of hours down the line. We really want to know what happens a couple of days down the line. Does tissue die? And one way to tell that is you could have an animal in which certain cells, microglia cells that sort of remove damaged tissue will sort of migrate into the region of damage. And they happen to have a fluorescent label, have green fluorescent protein attached to them. And this is 6 days later. And you can see you got this, have a cylinder of death. This is 500 microns. That's 500 microns. Okay, this is one vessel. So one vessel causes this damage. We have to understand this, both from a point of fluid flow and its health impact. Okay, so let me just skip ahead a little bit here because of time. One
line, Dave, is that you really wanted to take this understanding of this network, where its vulnerability is, and really try to connect this to disease like stroke, brain injury, really try to see what predictions it would make and do experiments to validate those types of things.
Yeah, I didn't want to be fast. I wanted to keep it on the physics here. I also don't want you guys to go running out of here saying I'm bored to death. But I'm just gonna say this in words. It's easy to say in words. The neurologists are a funny breed of people. They go, one vessel, it's irrelevant, right? 6 years ago, I went up to University of Washington and think of this guy's name. Every 5 minutes in this talk, this is irrelevant. This is irrelevant. Okay. Okay, in a month, the NIH is having a meeting on small vessel disease. So, in fact, see, if you use a small vessel in a visual area, your brain does this interesting psychophysical thing. It sort of fills in. So you can have an object that's moving, and you can't see it. But you won't say that, oh, I have a black spot in my visual field. You do this filling in. But you could do experiments that actually expose the loss of a small piece of neural tissue. These are experiments done by this former postdoc, Andy Shih, did fantastic work. And in a sense, if there's a specific sensory motor function that takes place in an area that died because you lost a single blood vessel, it's gone. And I think the way this relates to human disease is, it was just literally about a year ago, there's a lot of people in the medical profession who are trying to get high-resolution MRI of human brain going. Now, usually these are done at high magnetic fields, 7 Tesla, right? It's very, very high. It's been difficult to get it running, but there's a group in Belgium, Gert Biesels, and they really see these defects forming, okay? Even in human brains of people who are otherwise neurologically fine. Okay, so there's a chance that these things may just be happening. And you lose a microvessel, a capillary, no problem. You lose one of these penetrating vessels, you lose a little piece of your brain. So if one of your professors forgets the minus sign in Faraday's law, be charitable, right? It might be the wrong blood vessel, right? Just to sort of relearn it in another part of the brain. So what I wanted to do here is, I don't want to scare anybody, but we have this map, and we have this puzzle, and the puzzle is, why isn't blood flowing in from the side? Let's just sort of let's gain some physical intuition. So once we have these maps, okay, we basically just apply, Kirchhoff's laws, right? So we know at the top line is just saying at every vertex, right? I mean, the sum of the current flow has to go to 0, period, right? And I just write this for all, sort of 10,000 nodes. You convince yourself, after a week of chasing numerical errors, what you find is that you could actually solve for the flow in every branch. There's no free parameters in these systems because I know the pressure head on the vessels going in. And like I said, as I showed you before, I also know what the resistance is of all the vessels in this case. So let's just do the first bonehead calculation, which is on the computer, which is I take my measured vasculature, now reconstructed in terms of resistances, and I could just inject a numerical die, so to speak, and I could ask what the territory is, like what territory is filled up with half that die. And as a function of how much flux there was coming into that vessel, I could get an idea of the volume. And these are all these dots are just all different vessels from all the reconstructed vasculatures we did. And the diamonds here are the size of the regions of the brain that die, okay, after we make a blockage to one of these vessels. So not surprisingly, what this says is that the region that dies is the region that's primarily fluxed by a single vessel. So of a self-consistency. Okay, these are log scales, so this is sort of pretty rough and ready, but it kind of makes sense. But a true test would be to say what happens as a result of blocking one vessel. So Nozomi, now in her own lab at Cornell, went ahead and redid the experiments she did as a graduate student, but she did them in a much better way. So a fellow named Chris Yu, also former Bell Labs, right, sort of showed that there's a window, there's a water window where the absorption of water is very weak. This is at 1260 nanometers. And he built a laser that operates at that range. And the consequence is that Nozomi and Chris Schaefer were able to image now deeper into the brain. And they could map what happens as a consequence of blocking a vessel, not as a function of distance, which is very qualitative, but as sort of as a function of topology. One vessel downstream, two vessels downstream, three vessels downstream. And they gathered this data. Okay, there's a lot of scatter. That's just biology. It was actually a very hard experiment. And then what we did here, Pablo Blinder, now Professor Tel Aviv, Phil Tsai, who's been with me now for many years, is we took all our reconstructions and we did the numerical experiment. We blocked a vessel. And that's shown by these red dots. No free parameters. So the two just match. So somehow, an experiment that says this is what happens to flow when I block a vessel, and the calculation of that based on the reconstruct vasculature agree. So this is a self-consistency argument that possibly we didn't make the same mistakes in two different types of experiments. But it doesn't give us any insight. So let me just close with a little bit of insight. And so the exact reconstruction, these calculations give you this, capture the essence of what happens, but we want to get a little insight. We now finally want to put our physics hats on, and let's understand the essence of this in the simplest possible way. So let's do something that would just freak out any biologist. Let's assume a one-dimensional brain, okay? Just to get the essence, right? So this is a penetrating artery. That means there's flow due to a pressure head. So a pressure head means there's a battery. So this current that comes out of this artery, the resistor is the cortical resistance, is the vasculature. But we showed that no matter how complicated it is, the resistance goes to a constant. So it reduces to one resistor. So then there's an artery, and then that drains into a vein. And there's no pressure head here. This artery drains into this vein, and this goes on forever. Now, if we block an artery, just in this simple 1D picture, what's happening is that flow gets shunted by this vein, and flow from this side gets shunted from this vein, and this place gets no blood, okay, because of these shunts. These shunts have very low resistance, which is something we measured. So it's not that you couldn't push blood from the side. It's just that you have a low resistance set of drains. So now we can make a fancier model, right? So we make a two-dimensional lattice that respects a couple of things, a couple of bits of experimental reality. One is there's about twice as many veins as arteries, twice as many blue dots as red dots. The arteries and the veins have low resistance. They're kind of like hubs. So this is the vasculature resistance, all these little resistors. And now I ask what happens when I block an artery. And that's shown here. So obviously the real brain is not hexagonal and doesn't have all this symmetry. But this just sort of captures the essence that flow within this lattice just gets sucked up by all these different venules that surround it. It never gets into this region. Okay? And we just take this simple lattice model. And the red triangles, again, are the data that comes from Nishimura and Schaefer. And this is our lattice model. You know, it's not too bad. It's almost as good as a fancy computer model, except this you could actually do by hand. Okay? And what you also find is something rather interesting is that if you remove a venule, okay, which it seems tricky, like why would this happen? This is not where I'm going to get a clot, but this does happen during disease states. Then also, there's a region here that doesn't get any flow, and the little line here is the theory, but the blue dots again are data that comes from John Wynn in Chris Schaefer's lab. So the bottom line here is we started just asking a question: how does blood flow? Simple question. We had to use a lot of techniques that really came out of the world of physics, or just sort of take tools that were in the physics world but never had been applied to biology before and push them a little bit. And then you start to have some fun. And, but at the end of the day, you know, if you go on in physics, go on in physical science, what you want to do then is to take all this complicated data, computer simulations, blah, 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 and reduce it to something really minimal, right, that you could kind of get some qualitative understanding. All right. And that is, thank you. Okay.